Hello everyone. Welcome to my YouTube channel. I am Changlin Wan. I'm a PhD student at Purdue University Electrical and Computer Engineering Department. This is my first YouTube video. This video is made for the annual conference of MEBF Computational Biology and Bioinformatics Society, which is supposed to happen in the beautiful Cary, North Carolina from April 26th to 28th. This conference is the biggest annual gathering of computational biology and bioinformatics researchers in the Mid-South area in the United States. This conference also recognizes excellent young scientists, young and upcoming talent with Young Scientist Excellence Award. This is a very competitive award that only 3 PhD students will be selected as a finalist for this award and will give a preliminary presentation in front of a panel of experts during the conference. However, due to the COVID-19 pandemic, very unfortunately, this conference has been cancelled, and the Young Scientist Excellence Award becomes a virtual event for this year. I'm very honored and fortunate to be one of the 3 PhD students for this award. I will talk about my current progress on computational biology and bioinformatics, titled as Seeing Through Gene Expression with Novel Statistical Model and Data Mining. If you want to collaborate with me, you could find my personal website in the description below. If you are interested in the MCBIOS conference and the Young Scientist Excellence Award in the future, you could also find the website links in the description below. So first, let me give you a short introduction about myself. My name is Changlin Wan, you already know that. I was graduated from University of Science and Technology of China, majored in bioscience. During my college, I had several internship experiences in China, South Korea, Israel, and Rice University in USA. From these professional services, I realized my passion and the importance of computational biology and bioinformatics, which could really help us understand the biological mechanisms in a very bigger picture rather than just focus on 1 or 2 gene interactions. I was later admitted to the electrical and computer engineering program at Purdue University as a PhD student. While at Purdue University, I have systematically studied the statistical and data mining methods. So on the one hand, I'm a YSEA PhD student, but on the other hand, I have a diploma of biological science. So I managed to integrate what I have learned throughout those years to apply statistical methods and data mining algorithms in analyzing high-dimensional biological data like single-cell RNA sequencing data. I was very lucky and fortunate to have Professor Chi Zhang from Indiana University School of Medicine as my advisor. And just several months ago, I have declared my candidacy for PhD. So without further ado, let's get to my presentation. As we all know that cell is a basic structure, functional, and biological unit of a living organism. This figure highlights the process of gene expression in each cell, from the unfolding of DNA sequence to transcript into RNA, then translated into protein, where the protein carries the functionalities. With the development of modern sequencing techniques, we now have the power to sequence every information of gene expression in single cell. First, we could get the single-cell genome sequencing information using SNS or SCI-seq. We could derive the DNA histone interaction by single-cell ChIP-seq and single-cell ATAC-seq. We could also explore DNA methylation process by single-cell MAC-seq, identify mRNA by single-cell RNA sequencing, and identify the surface protein by stat-seq or rip-seq. Among all the techniques, single-cell RNA sequencing is the most popular one for two reasons. First, mRNA is a messenger that links DNA to protein, which means it carries the most significant information of an individual cell. Second, single-cell RNA sequencing is the most mature and robust techniques in the market. Its robustness and efficiency is mainly driven by the introduction of two most important protocols by far, the SMART-Seq2 protocol and the 10x genomics protocol. Single-cell RNA sequencing granted the unprecedented resolution for us to characterize each cell. The most common application is to differentiate cell type within a complex tissue. In general, we could get an expression matrix of thousands of cells with thousands of genes. Then we could apply dimension reduction techniques like PCA, t-SNE, or UMAP to put each cell into lower dimension. Then we could apply the clustering analysis methods like k-means, spectral clustering, or community detection algorithms to get different cell clusters related to different cell types. This analyze process is what I regarded as cell-wise perspective, in which we could get the cell clustering, we could identify different cell types, and analyze the components of each sample. However, there is a fundamental flaw in this process. That is, even we know perfectly clear about the cell type, we still do not have precise understanding of how each genes behave in these cells in the complex tissue. This is another perspective, the gene-wise perspective. We could use a single-cell RNA sequencing data to infer the gene activities, and we could even derive biological mechanisms underneath the expression data. That is where we come to play. We take gene expression from all the cells and inference different expression state by our LTMG model. We claim these different expression states are resulted from different regulation of gene expression, which related to different biological events inside a cell. LTMG is short for left truncated mixture Gaussian distribution. It has three major components. AE as active expression, where each AE peaks related to different expression states. SE as suppressed expression. This peak alone stands for the noise inside the data, and the Z-cut is the minimum detection resolution for single-cell RNA sequencing, where the expression value above the Z-cut can be identified by single-cell RNA sequencing, but the expression value of below the Z-cut cannot be identified by single-cell RNA sequencing techniques. This model has been published in nucleoside research. Out of brevity, I'll omit the mathematical derivation of LTMG model in this presentation. But if you have further interest, I also attach the paper links in the descriptions below. So we first test whether LTMG can truly fit the data. Here we use Comograph statistic test. This test measures the distance of fitted distribution with real data. The lower the value is, the better the fitting will be. We tested LTMG with three state-of-the-art distributions: ZIMG as in zero inflated mixture Gaussian distribution (MUST), and BPSC as in beta-Poisson distribution on over 80 real single-cell RNA sequencing datasets, shown in the right figure. On vast majority of the data, LTMG achieved the most stable and better performances compared with other other 3 tools. We also benchmarked LTMG with other 10 distributions in a paper published in BMC Bioinformatics. LTMG remains its superior performance as well. Next, we validate whether each component of LTMG carry information. First is the SE peak. This peak contains noise in single-cell RNA sequencing data. We give an example of GSE 103322 dataset in right figure to validate that cells that are in SE peak is not true expression. In figure D, we select 8 T-cell genes and 8 fibroblast genes. The color measures the T-cell or fibroblast cell enrichment in AE peak or SE peak. Shown in the figure, most cells express gene markers in SE peak are not T-cell or fibroblast cells. We give the cell distribution on granzyme K, the T-cell marker, and the collagen 6A3, the fibroblast marker, in figure E and F. As you can see, the SE peak indicated by the green curve is mostly constituted by other cells that are not supposed to express T-cell or fibroblast marker genes. We also give you the cell distribution on the t-SNE plot. Other cells in SE peak did not result in the same cluster with T-cell or fibroblast cells. These results validate that SE peak can accurately detect the noise in single-cell RNA sequencing data. This could serve as a denoising process in analyzing single-cell RNA sequencing data. So, SE peak could denoise the data, but more importantly is whether the AE peak truly related to biological events. That is to say, whether the AE peak truly reflects what's going on inside the cell. This idea is very hard to prove thoroughly, but we managed to find some clue. Let me first introduce the single molecular fluorescence in situ hybridization, also called SM-FISH technique, also called SM-FISH. This technique, shown in the right figure, could visualize gene expression under the microscope, such that we regard this data as a gold standard of gene expression in single cells. So we managed to get one dataset that has both SM-FISH data and single-cell RNA sequencing data from the group of Professor Arjun Raj at the University of Pennsylvania. We fit our LTMG model and other two models on the single-cell RNA sequencing data across 20 genes and test the KL divergence of the fitted distribution to the SM-FISH data. The smaller the KL divergence is, the better the distribution fits the SM-FISH data. As shown in the right figure, LTMG showed constant better performance than other methods, which proved that compared with current approaches, LTMG can better reflect the biological events inside a cell. So, one of the important benefits to use LTMG is that LTMG increased the interpretation of single-cell RNA sequencing data. LTMG transferred the single-cell RNA sequencing data into discrete expression state. And if you use a state value to conduct the low dimension visualization like t-SNE, LTMG gives you a much clearer results compared with just using t-SNE or similar on the original dataset. So a quick summary here. LTMG is the first complex model to characterize gene expression behavior. It could denoise a single-cell RNA sequencing data that identify the expression state is indeed related to biological events. It also gives you a better low-dimensional visualization and increases the interpretation of your data. Overall, LTMG is a valuable framework it could help understand further of the complex mechanism in tissue and organisms. So LTMG could transfer gene expression data into discrete gene expression state data. The next natural question is, how could we identify gene expression patterns across different cells, indicated by the different colors here? Here we introduce a classic data mining method called binary matrix factorization. This binary matrix factorization could identify a set of cells express the same set of genes, which could help us determine the cells with the same functionalities and the genes that actively interact together in the complicated biological events. So, however, binary matrix factorization is an NP-hard problem. What the NP-hard means is that we could not have a very fast and memory-efficient algorithm to get the best results. So here we propose the MEBF algorithm. We focus, since we could not get the best results, we focus to get the rather optimal solution under the geometric consideration. This algorithm has been accepted by AAAI 2020 conference. The link for this paper is also in the description below. We compared MEBF with 3 state-of-art Boolean matrix factorization algorithms. This figure showed the time consumption of MEBF with other methods. MEBF is on average 10 times faster than other methods. MEBF also achieved better results than other algorithms in reconstructed error and sparsity in matrix factorization. Due to the time limitations here, detailed results and explanations could be accessed in our original paper with a link below. So, one of the important applications of the two introduced algorithm is to identify the functional cell clusters. Take this head and neck cancer cell data as an example. By applying current analysis methods, we could identify different cell types like fibroblast cell, T cell, endothelial cell, etc. But we could not identify cancer cell clusters marked in the yellow here, because all the cancer cells are clustered to different patients, means each individual patient has different cancer cells. This is a rather bad news, because if all the patients are different, it provides a huge problem in design treatment for each individual patient, and also the treatment can become very, very expensive. So by applying LTMG and MEBF, we get quite different results. We still have some cancer cells clustered to different patients, but as you can see the circles in the figure, we managed to get 2 cancer clusters that are mixed with multiple patients. We then take a deeper look to these clusters. We identified these clusters have distinct behavior in cancer metastasis, especially in EMT process. The mixed cancer cell clusters are in M stage, while the patient-specific cancer cell clusters are in E stage, which indicates that cancer cells may arise differently in their original tissue, but when it comes to metastasis, they will share great similarities, or it could be they have limited solutions to metastasis. This could become some potential target for us to design general treatments for all the cancer patients. So, in summary, we provide a cutting-edge analysis platform to sift through the gene expression data. The proposed LTMG model characterizes gene expression behavior. The MEBF binary matrix factorization algorithm derived the expression patterns. Together, we identified functional cancer cell clusters across different cancer patients. Noted this platform can smoothly extend to any single-cell dataset. It increased the interpretability of single-cell data. This platform could also push our deeper mechanism-driven understanding of biological research. With that, I would like to thank everyone in the Biomedical Data Research Lab at the Indiana University School of Medicine. Professor Chu Zhang and Professor Sha Chao has been my wonderful advisor. Wennan, Xiaoyu, Peng Tao, Jiaming, and Alex gives me continuous support. I would also like to thank my collaborators at Purdue University, Rice University, Amazon, and Ohio State University, and UT Health. Thank you so much for watching this video. I'll continue to generate some exciting introductions of our work in my YouTube channel. If you like it, please thumb up and subscribe. Thank you for your support. If you want to collaborate with me, please access my contacts in the description below. And also, if you are interested in MCBIOS and the Young Scientist Excellence Award, you could follow up on the information below. Okay, thank you so much, and see you next time.